Thank you, Richard. Thanks to Dr. Choi and the Chinese University of Hong Kong to invite me and to make this possible. So what I want to do today is just give you a little bit of a taste of what is dealing or handling children with undiagnosed rare disorder, which is something that is almost part of my everyday clinical practice. Let me see how I move this. Here we go. So there are a lot of definition about rare diseases, but essentially we now have approximately more than 6,000 rare diseases. And we know that the numbers vary, but it is estimated that 3.5% to 6% of the population have a rare disorder. So even though these are very unusual, infrequent conditions, but altogether they represent a really large number of them. So in the U.S. and also, you know, in Europe, is approximately with conditions that have an incidence of 1 in 1,500 or 1 in 2,000, and also, if you take it as a whole, about, you know, a prevalence of less than 200 people in the U.S. So again, rare, but not so rare after all. So when you're trying to, you know, look at these conditions, so essentially these are conditions you have probably many patients that have come to the clinic many times over the years, and there's still not an answer. So they escape, you know, a diagnosis even for the most capable physicians and clinicians. So oftentimes some of these disorders affect multiple systems, and they also represent a significant, you know, chunk of what the healthcare system has to deal with, because all of them having multiple medical problems, and that has a significant burden in the system. Not all, but many of these rare disorders are genetic in origin. There are a lot of really things that may be related to toxic exposures or immune disease, but a large number of these are in the genetic realm. According to NORD, which is an organization that deals with rare diseases in the US, an average of 5 to 7 years is needed to have a diagnosis in some of these patients. So, and when it comes to genetics, you know, we have really gone from the basic karyotyping back in the 1960s to more molecularly involved techniques like FISH, or fluorescent in situ hybridization, that started in the 1980s. And very soon after that, and thanks to the Genome Project, you know, you had all different types of testing that were now becoming available to the public, initially in the hands of researchers. And that goes for chromosomal microarray, whole exome sequencing, and even whole genome sequencing, that, as you know, is something that is widely used in the clinic. So beyond that, there are a lot of other tools that can be used, and that is, you know, going now in the omics, like transcriptomic, dealing with RNA sequencing, or metabolomics. But when it comes to the research arena, one of the tools that are becoming very important are working with animal models, and that's whether you're working with mice, zebrafish, Drosophila, etc. And there is a lot of now expertise about how to introduce genes and knock down genes in these animal models. That helps a lot with the investigations, and a lot of, you know, other things that are coming to the clinic. Long-read sequencing still in the research, but probably something that will come to the clinic, and things like optical genome mapping. So obviously, all of these techniques is about trying to decipher the human genome, that, as you know, is a large amount of DNA that composed by 3 billion letters or base pairs. We have approximately 20,000 genes, and currently, we know that 6 to 7,000 of them are associated with disease. So obviously, the DNA is just one part. It's just looking at the genetic code, and we will be talking about some of the other technologies in a minute. So one of the techniques that have been used more in the clinic is something called exome sequencing or whole exome sequencing. And and now we're moving on to the genome sequencing. So the exome sequencing essentially captures all of the exons in the genome. You know that the exons are the part or the coding part of single genes, and the exome allows capturing of those particular exons, sequencing them in depth, and hopefully coming up with an answer to diagnose your patient. About 85% of the disease-causing changes happen to be in the coding regions of the genome. So the human genome or the whole genome sequencing is obviously a bit different, because now you're looking at every single thing and every piece of the DNA in the organism. And obviously now all of these is looked at and compared to consensus and standard references for normal with normal, comparing it to normal individuals. So this is something that I have shown in the past. So essentially what you have is on the left, you see your DNA, and you have exome versus genome. And in the case of exome, what you do is you break up your DNA fragments, and then you use these repair ends and add the adapters, and then only capture the fragments that contains the exons. And then you will essentially wash them and then start sequencing and aligning them. Just keep in mind that all of these are done from small segments of DNA, and that is what this technique is all about. Ultimately, you do the variant calling, and then you start making your diagnosis, hopefully. With regards to the genome, you essentially go to these steps of putting the adapters, but essentially you just sequence everything, and not just the exons that are enriched in the exons, exome, but you just do look at absolutely all of the genetic material. And also, you have the transcriptome. So essentially, the transcriptome is looking at the RNA that is copied from the DNA. And essentially, what you try to find out is what happens with the DNA when you have a particular variant or change, and whether that will affect in the way that RNA is being made, that essentially will be translated into a protein. So Wes has some strengths and also has some pitfalls. So the strength is a very efficient method. You can do look at thousands of genes and thousands of exons all at once. And once you make your discovery or your analysis, hopefully you can identify medical relevant or actionable results. And as I mentioned, it's the well-known part of the genome. It's less expensive. The data collection happens to be much easier than doing in genome, and it takes much less interpretation time. So West, however, has some limitations, and the limitations are that the exome study doesn't cover 100% of the exons. So it's almost 90 to 95%, and there are some regions of the exome that are very difficult to interpret because of repetitive sequences and areas that are very difficult because of the DNA context to be sequenced in detail. What the exome doesn't give you is if you have either large deletions or duplications, you may be able to get like 2 or 3 exon deletion, but you may actually miss a very small deletion, or you can even miss very large deletion, and the same as duplications. Deep intronic mutations is something that the West doesn't get. Regulatory mutations or things like mutations in untranslated regions of the genome. So you have some difficulties on getting some diagnosis that you can actually achieve with the genome sequencing. Then this is just something I'd like to compare. This is a single exon from the APP gene. This is a gene that is associated with Alzheimer's disease. And here you see a coverage by the exome over that particular exon. And you compare the same view, but now in the coverage for the genomes. Then I think the genome covers everything, but also sometimes you can miss some of the depth that the exome can have. If you see there are a lot of overlapping, segments that are reading this particular exon with the whole exome sequencing, with the genome, is covered, but it's not covered as well, not covered as many times. So, and in terms of the comparison, obviously, most of the genome is difficult to interpret because most of the variation happens there, and most of the variations are non-coding regions. So it's always complex to try to make calls in the non-coding elements like the regulatory regions. So oftentimes there is not as much experience, and it's difficult to determine pathogenicity. And in the genome, you do have a lot of non-coding RNAs, and most of the times they don't have much of a role, but in the past few years, there's some long non-coding RNAs that have been associated with disease. So it's something that we still do not understand as well. And certainly over the years, as we get better, we may have more diagnosis based on some of these calls. Now let's go to the detection rate. Well, the detection rate has varied, and because the exomes and the genomes have also become better. In the beginning, we will do a whole exome sequencing in single individuals. That's called proband exome. And the range was approximately in the 20% range, 21, 22. Some studies even up to 31 as the highest. Having said that, when these studies were switched from proband to trio, meaning using the sample of an individual and their parents, the reporting, the detection has become better, and it's definitely over 30% and up to 37% in some cases. Genome is a little bit different, and this is from a study that was done several years ago, but these were done on neonates that were, essentially sicker or ill infants in the neonatal ICU. And in this particular collection, the detection rate using genome was 57%. I will say that by most part now, you look at all of the different studies, genome detection rate is around 50, maybe a little bit over that. Perhaps this is a different sample because, again, you are dealing with very sick children. And one of the things that now is coming online is the use of long-read sequencing. So when you do next-generation sequencing and when you do exome sequencing, what you will find is that all of these segments that are broken down of the DNA, they are short segments, and they are typically sometimes not enough to cover certain very complex areas. So, and you go from 100 to 200 bases to sometimes several kilobases. So the long read sequencing actually is been very helpful to detect areas where they're more complex because of the DNA contents, and you can even now do you can look at some unusual situations where there could be inversions, there could be some rearrangements inside a particular gene, and also, you know, it's a very good technique to look at phase when you don't have, you know, parents. By phase meaning you have a couple of mutations and you don't know whether they're in cis in the same chromosome or trans in opposite chromosomes that will be associated with disease. So what comes next? Obviously, and what do we use for clinical and for research? And then ultimately, that's going to turn clinical. So exons and genomes, genomes now are gaining a lot of popularity, are becoming some good tools. The advantage of a genome is that you can look at all of the sequence of the DNA, and also you can look at copy number variations, which is not something that you can do with the exon. But again, and I will show you examples, sometimes you fall short. So, you know, now some of these new tools are looking at the RNA, looking at expression, looking at the sequencing data, and see whether that is causing any problems in the readout, and using metabolomics and animal models. Long-read sequencing are being used, but it's something that is still very expensive and that requires fairly special equipment. So not many labs are, at least not in the US, that can perform these studies. I'm sure probably Asia is different. So this is a long slide, and I apologize for that. But essentially, in a study that we've done, and I'll tell you more about the UDN, this is the Undiagnosed Diseases Network that functions in the US. In our group, there was a study looking at transcriptomics in some of these UDN cases, and it was estimated that there was an improvement based on our data of diagnostic rate of 12% from the entire cohort, or 17% after the cases that were excluded from genome or exome alone. So essentially, you're adding at least 12%. And there are some examples, you know, that we have shown. I'm not going to show those to you today. I will show you other cases, but those were people with Cooling debris syndrome, Rempenin syndrome, TBCK, encephalopathy, and other genes associated with intellectual disability. Transcriptomics can get a little bit tricky because sometimes you require different type of tissues, and most people will do studied normally on blood, but when it comes to transcriptomic, sometimes you need to do skin fibroblasts just because of the expression on many of the genes. If you're looking for blood-related disease, yes, certainly a blood sample may be useful, but you need to have at least a good expression of 10 parts per million if you want to do transcriptomics. And the Undiagnosed Diseases Network, we are now in year number 10. It has 2 rounds of 4 years each and an extension, and now it's probably going to be refunded by the National Institutes of Health in the US. It started as a place with 4 sites, and now they're up to 10 sites, and there's some now affiliated sites. In addition, there is their core site, so there's the genome a site that is actually at Baylor as well, and there's some core labs like Drosophila, zebrafish, and also mouse modeling. So it involves a lot of different people across the network. And this was modeled after a program that was started by the NIH called the Undiagnosed Disease Program. So they did it internally, they still do it internally, and they sort of exported that to a larger scale to these UDN and diagnosis network. So I will show you 3 different cases to highlight some of the importance of these particular studies. The first is for a patient that was referred to our group that I saw in Houston. This was a young boy from San Antonio that had presented with a low bone density, bowing of the femurs, mild delays, especially in the growth motor area, lot of different fractures that happened with no significant trauma, blue sclera, no family history, and the testing has been negative. Essentially, the prenatal and birth history were completely unremarkable, born to a young mother, normal ultrasounds, normal growth, born by cesarean section at 39 weeks due to heart rate decelerations. The birth weight and height were in the normal range. Blue sclera were noted already early in after he was born. Developmentally, he pulled to stand at 9 months, saying words at 12 months, walking at 15 months, and starting to put words together at 18. At the time that he was first seen, he was in pre-K and was having regular education, having no problem. So he was seen initially by a geneticist because at 3 years of age, the orthopod or orthopedic doctor suspected he had OI type 1. In his lifetime, he had a history of 5 different fractures. The family history is essentially negative. That has keratoconus. The radiograph shows that he has significant osteopenia, and the exam had the blue sclera and also darkly mottled teeth. All of the labs done for bone metabolism were normal, and the diagnosis of OI was suspected. A skeletal survey showed a lot of changes related to the osteopenia, multiple growth recovery lines that, the recovery lines are lines in the skeleton that usually happens when some particular disease events, if they're not recovery, actually they have to do with something that happened at the time and typically metabolic. Liver was enlarged. There was mild bowing of the femurs, and also the X-ray showed that the tibia was previously fractured and now healing. And he had a few Wormian bones. Wormian bones, you may know, these are like small bone islands that you can see in the skull. can be seen normally sometimes, but is more commonly seen in patients with osteogenesis imperfecta. The exam later on showed the blue sclera, hypermobile joints, the muscle bulk was decreased. There was some unusual streaky pigmentation over the limbs. He had an awkward walking gait and had some difficulties running and had normal hearing. So these are the growth charts. These are the height, essentially around, a little bit over the 50th centile, now going down, but still around the 50. And the growth has been on the short, on the small size. So what happens to this kid? Well, genetic testing was done, and some collagen and specific genes for eye were done, and those were reported as normal. Then a panel testing was requested to include more genes. This is a panel done by Emory University, and that was reported as normal. Whole exome sequencing done by GeneDx was normal. So the UDN accepted, this child, and typically what is done is the West is just to make things a little bit less expensive. The West is recover the data, is reanalyzed by the research group, and if nothing is found, then they move on to do the WGS. And we also have the trio whole genome sequencing. So now comes the interesting part. So the trio genome was reported as normal until the RNA was done, and the RNA showed some findings that were interesting. So the trio was revised and then found that indeed there was a mutation, an intronic mutation, deep intronic mutation in COL1A1, which is the gene associated with osteogenesis imperfecta, one of the common genes. And an intronic abnormality is going to cause quantitative defects in COL1A1. And that's what happens in OI type 1. So it turns out that when they did the RNA sequencing, the RNA showed these cryptic acceptor site mutations. So essentially, what you had is you have this abnormality that was causing by this intronic mutation, and therefore leading to an abnormal exon. And why was this not picked up clearly early on? Because this was only present in 10%. So this is a 10% mosaicism for a deep intronic mutation. So you cannot get any more complicated, but it does explain very clearly what happens to this case. So and again, this mutation was found after this splicing abnormality was seen. So imagine all what it took for this patient that had a classical diagnosis of OI to be diagnosed. And clinically, yes, you look at this case, you look at the history. This is OI. This is a mild form of OI, and see how many years it took until someone was able to demonstrate. Does it matter? Well, it does matter, because now the patient can be treated accordingly and also can be counseled accordingly. And if he's to have children when he grows up, certainly you know what to look for. So this was an interesting case. So in summary, this is a COL1A1 deep intronic mosaic variant, consistent with OI. So we believe that this is our diagnosis. So do we know how many of these situations happen that maybe there are small mosaicism for some genes in the organism? Is possible, because some very, very tiny, sometimes some of these abnormalities in splicing can be seen in regular tissues in regular people. But we think that the 10% splicing is probably enough to explain this disease and this child. And then one other thing is that we put this particular gene mutation in gene matching. Gene matching is something that is available to everyone that eventually puts the information on one particular gene to see whether it has been reported. In this case, this is an intronic variant, so it will be interesting to see if someone else did. And it turns out that we got responses from 4 different people, exactly the same mutation, the patients that have been described with osteogenesis imperfecta. So obviously, we're more likely going to be on the right side when it comes to the answer. I don't know if anyone has any questions. If not, we can leave those at the end. And today is all about bones, so I apologize, but everything happens to be with the skeleton today. So this is a patient that has been seen in the clinic for many years. This was a patient of Dr. Brendan Lee, and now followed by Dr. Sutton. This is a patient that has a history of multiple fractures, has bilateral sensory neuron hearing loss, and other problems that included delayed puberty and ASD. So this patient had been followed for many years, and no one ever was able to diagnose her. This patient even had many years ago collagen analysis on fibroblast, and that was normal. So these are some a bunch of different X-rays that go from very young age to more adult age that shows some of the gross abnormalities that you see in the bones secondary to fractures. And look at these right humerus that has a lot of deformity, all associated with fractures that happened in the past, and very severe kyphoid scoliosis at more adult age when she was 16. So interestingly, this patient was found to have a mutation in a gene that now, it was a gene discovery in a gene called KIF5B. And this is actually a gene that codes the heavy chain of a unit of a protein called kinesin 1. So this kinesin 1 has a significant role in development of muscle, nervous system, cartilage, and adipose tissue. So what is believed, like the kinesins are all motor proteins. So essentially, they deal with movements of different proteins moving across the cell. And they are present mostly in secretory vesicles and also expressed in receptors. And they're very commonly found in the mitochondria. So the role is pretty wide. They're in the Golgi, they're endosome, and they are associated with cilia biogenesis and function. So we have known also the kinesins are associated with different disorders. In fact, there are other conditions associated with abnormalities in kinesins in neurodevelopmental disorders, in patients that had multiple anomalies, including renal dysplasia, polydactyly, and hydrocephalus, sort of like as you see sometimes in ciliary abnormalities. And there is a skeletal dysplasia, it's called SEMD, which unlike CT type 2, that is also associated with echinacin. So at the point that this patient was being studied and this was found, some of the people in the core lab started trying to doing some modeling to see whether these particular defect caused disease and whether this was the responsible one for the disease. So what they did is that they identified an ortholog for this particular gene in worm. They knock it down and they introduce the mutation that is now known as T90I. So when they did look at the wild type and the controls before they introduced the mutation, these worms were fine. Once they did knock down or had models for homozygous mutations or deletions, it was found that all of these, the larvae, actually did not survive. So it was believed that this may be a different mechanism. And so when they look at the variants are, looking at the worms. So these are the normal worms, so you can see the normal movement, but these were actually the ones that had introduced the particular mutation in the counterpart for this particular model. So compared, how slow is the movement of this particular worm in question. So when they look at the swimming speed in the heterozygous, they immediately found this was the wild type. This was the one that had the mutant and change, and this is the one that has the heterozygous mutation, so the T90I. So basically, this looking at the swimming speed was severely compromised. So there were the heterozygous display dominant phenotypes not observed in deletion heterozygotes. So it was believed that these, the T90I was a gain of function mutation. So once they did added some extra copies of wild type alleles, they were able to show some recovery on the phenotype. So ultimately, it was believed that some of these extra copies of the wild type were able to rescue. So that's why they concluded that this was a dominant negative allele. So in conclusion, these models, that they have, and there might actually other experiments that I'm not showing you, that these heterozygous mutants had fewer moving mitochondria, decreased velocity, and decreased distance. So, and they have inability to move the mitochondrial cargo along the microtubule. So the idea is that this modeling can show that this kinesin mutation has an antimorphic or dominant negative behavior. So this is essentially the summary, and based on this particular type of experimentation, it was shown that indeed the novel mutation in this patient was likely causing the OI. So obviously it was a single patient, but now you have functional data, and the functional data is showing you your answers. So another 4 patients were later on published. They were identified by other groups, and all of these patients have mutations in this KIF or KIF5B, and they all were de novo, and they were all associated with a fairly form of osteogenesis imperfecta with multiple fractures, scoliosis, all very similar to that was seen in our patient. So in conclusions, these heterozygous mutations in these KIF5B genes were identified in 4 additional subjects with OI, and the studies done on C. elegans, so the worm, show that this variant is damaging and likely causing disease by a dominant negative mechanism. There are other cell studies showing that there was an abnormal Golgi distribution and abnormalities that suggest a problem in intracellular transport effect. So this is probably one of the first time a protein that is not necessarily dealing with collagen or chaperone protein for the collagen are associated with osteogenesis imperfecta. So, and the last one, I think that we still have a little bit more of time. This is from a case that I was involved in the first few years of the UDM project. This is a patient that I have followed for many years. She presented with severe failure to thrive and short stature. She also had severe neutropenia and required these granulocyte, a factor to maintain her neutrophils. She also had gamma globulinemia and basically didn't mount any antibody response when given a vaccine. She was born in Egypt, and the family later on migrated to the US. And she was already diagnosed with neutropenia and has 2 normal siblings. Nothing much to say about the pregnancy, but she was already noted to be small, and she was slightly premature, and she was small for length, weight, and head circumference. This is a patient of this cute girl at 5 years of age, and she was essentially completely normal. There was nothing really unusual about her. She had this clinodactyly of the 5th digit, and this is the growth chart. So this is where she's growing, and obviously compared to a normal growth curve. So X-rays that were done on her show some flattening of the vertebral bodies, and she has some metaphyseal changes. So you can see this ragged appearance of the distal femurs that suggested a metaphyseal dysplasia. So at the time, exome sequencing was done, and then later on was studied by the research group, and this child was brought for a new evaluation. And there were 2 variants that were reported on a gene called tonsil, TONSL, and these mutations were missense mutation. They were not present in exact or nomad. Obviously, I have probably very old numbers. Nomad has a lot more cases than these. And the CAT score on these two cases, one was on the high end, and another one was very high. But that's the only information we had up until we learned about two siblings that were in the US that were found to have similar mutations in this same gene, tonsil. This is the boy. He started with congenital neutropenia, history of multiple infections, and had more than 20 hospitalizations. So you can get the idea of the degree of his illness. Had very low immunoglobulins, IgGs and IgM, and had neutropenia with ANC of 0.9. So he has been receiving immunoglobulins and also GCSF. So the growth on this child wasn't that significantly abnormal. And here are the X-rays. So you can see the vertebral bodies that are abnormal. They're short, they're flattened. They have almost like a central depression. And the X-ray shows this sort of unusual striation. So if you see almost, they look like lines towards the end of the bone, but they're very subtle. This is this boy's sister. She was at the time 10.5 years, and she was born full term, no problems, normal weight, also diagnosed with neutropenia and hypogammaglobulinemia, multiple infections as well. She was on the short side, although at the 11th centile, also with a low ANC and low natural killer cells and IgGs and IgM. So very similar to the brother. Here you have some changes on the spine, not as much. Maybe a little bit of flattening on some thoracic bodies, and perhaps you can be talked into say that there are some lines, and I will get to the lines in a minute. So the siblings were found to have also compound heterozygous abnormalities in the same gene, tonsil. One was a missense mutation that was transmitted by the mother, and the other one was an intronic mutation that was transmitted by the father. So this is actually the proband fibroblast of one of the proband, and there was essentially a retention of these introns, intron 7 and intron 8. So here you have, sort of for you to familiarize yourself, this is the intron 8, exon 8, and intron 7. This is the control fibroblast RNA, and you can see the exons, and then you can see the intron regions. But in the proband, you see continuous lines. So essentially, you have these introns being retained in the cell. So as it turns out, tonsil was now found to be causing a bone condition called spinastrium dysplasia. And these patients present early in life with short stature. They may have some dysmorphic features, mid-face hypoplasia, depressed bridge, picantal falls, prominent forehead, anteverted nose. They do have also short dental roots, and some of the radiograph will show some abnormalities in the lumbar vertebra. And as they get older, they may do show these striations or sort of lines that go along the bone and the metaphysis and irregularity of the metaphysis. So this is actually taken from an atlas, a very well-known atlas used by people in the skeletal dysplasia field. And you can see, probably here, you can see more distinctively these sort of like dark lines that go over the metaphysis. And you can see some irregularity of the metaphysis. And similar, you see a patient that has a smaller vertebral bodies. So there was a group that had some additional patients, and these patients were reported, and there was another group in Korea that had a similar discovery. So they were presented in two back-to-back papers in the American Journal of Human Genetics. But here you can see some variations. Some of the dysmorphic features I was telling you about, this unusual nose and mid-face hypoplasia and diverted nares, dental abnormalities. And this is from our publication with all of these other patients that were showing the different comparison of the bone findings in the vertebral bodies and in the legs. So what is tonsil? Tonsil is essentially a replication-associated protein that is involved in stabilizing these situations where you have DNA repair and DNA excision. So essentially, when you have loss of tonsil, you have increased levels of S-phase associated DNA damage, and these patients now get hypersensitive to certain agents, so like topoisomerase inhibitors, so which is actually something that has been used to sort of replicate some of the problem seen in the cells of these patients. So there were some initial models, and this is actually one of the first things that we have tried when we initially started working with these patients, doing the mouse model. They took a long time, but essentially was little in tonsil, so it wasn't really a good model to study. When we had these additional cases to study, we ended up getting collaboration from the group that doing zebrafish, and what they do is they created these models. They didn't introduce these mutations, but they essentially knocked down the tonsil gene, and they actually showed that these zebrafish had very significant decrease in the length, in the overall size, and also that they have a way to look at some of the neutrophils that were shown that have been significantly reduced. So this was sort of a good proof that this particular gene is involved in growth, and it appears to be very important in blood proliferation, especially for neutrophils. So these were experiments to look at RAD51, which is also a big component in the repair mechanisms, and these were from the wild type, and showing the RAD51, and then showing the different patients that have a significant decrease in RAD51. They also these patients show that had increased significance in spontaneous replication and forced stalling when they were exposed to CPT, which is calprotectin, which is a topoisomerase inhibitor. So that was further proof that these cell lines were more susceptible to CPT action. And then there was also another study showing that these tonsil, wild-type tonsil, could rescue this deleterious effect of these particular cell defects. So I will probably move on just to the aid. So I don't want to get too technical, but essentially, this was a situation where we went from a patient that was totally undiagnosed. We were lucky enough to find other additional patients. And as always happens, when you find something like this, you learn that another group is working in the same gene. So tonsil was ultimately put on the map. And tonsil is important for cellular viability. So the mutations cause significant disruption in DNA repair and associated with defective homologous recombination. Mouse models were not very useful, and that's why we were for 2 or 3 years with no answer up until we got better ways to look at the genes and other additional patients. And now we know that this phenotype is associated, tonsil patients have these condition associated with short stature and growth restriction. And obviously, even though the patient I had early on showed severe growth restriction compared to patients with phonostring, but there has been one additional patient reported with very significant growth abnormality similar to the one we have seen. And obviously now we were able to look at zebrafish and look at the knockout models to recapitulate the phenotype in a way, and to look at the neutrophils as well, production deficit. So in conclusion, whole genome sequencing has added a lot of additional information for the discovery of rare disorders. The caveat here is like, this is probably the first patient is one of many that I have shown in the past, where even the whole genome sequencing has missed it. Because sometimes has to do with the pipelines. The pipelines are not necessarily looking at everything. There has too many things to look at. So you try to select what are the most important, but ultimately, when you go and you do RNA studies, if you're lucky to have that, you sometimes may be able to go back and say, well, have I looked at this gene in particular, in the way I should? And this is one more of the examples that I have of patients that were missed, obviously by exome and then by genome. And then, we go back to that after, getting RNA sequencing data. So using RNA is certainly a way to help with the increase in the diagnosis. Obviously, not all mutations will cause transcriptomic abnormalities. So, if you're lucky that you have an expression difference in the particular gene because of gain of function, loss of function, or you have a splicing abnormality, you may be able to see something, but certainly, it doesn't apply to all mutations. And highlighting also that these 2 examples of osteogenesis imperfecta and now the tonsil that has essentially led to diagnosis of conditions, one that was known but had no gene, and another one that is a new form of OI. So this takes a lot of work. These obviously I'm not going to acknowledge everyone because it's a large group of people. I'm just one simple clinician in the whole group. But it shows that a lot is going on. We are going from undiagnosed to rarely diagnosed, and hopefully we'll move on to get more patients, and so we can help with diagnosis and essentially with treatment.